Thanks very much, Elliot. So my research is focused on understanding how rare mutations in genes can cause Alzheimer's and Parkinson's disease. And tonight I'm going to talk to you a little bit about some of the challenges that present themselves trying to understand what causes Alzheimer's, what causes dementia, and some of the advances that have been made in the past several years through some of the work looking at genetics. I'm going to start this talk going back over 100 years. This is a lady called Augusta Dieter. She was the first person to ever be diagnosed with Alzheimer's disease by our Alzheimer himself. Augusta was a patient in an asylum in Frankfurt in the very early part of the 20th century. And Alzheimer went into the asylum and interviewed her just before her death in 1906. What he found was a lady who'd withdrawn into herself. She was very, very confused at the world around her and found it very, very difficult to communicate with others. And I always think, looking at some of the photos of Augusta, that you can almost see some of this in the images that survive from that time. She has a sort of listless gaze and seems almost to be an air of sadness. And I think that's something that people who know individuals with Alzheimer's or maybe have family who have Alzheimer's can really relate to. Now, after Augusta died in 1906, Alzheimer was able to carry out an autopsy and look inside her brain to try and see what it was that was actually causing her to have these symptoms that caused so much distress. This is Alzheimer shown over here on the left. What he found was quite shocking. He found that the brain had almost wasted away, it had shrunken within the skull, and that this was due to the loss of lots and lots and lots of her brain cells. And he found that in the cells that were remaining, there were accumulations of sticky protein, lumps almost, within the cells and around the cells outside. And these are actually very, very similar to what modern neuropathologists see within cells as well. Now, this is all over 100 years ago. And in many ways, it's incredibly frustrating that in all that time, we haven't been able to develop enough of an insight into Alzheimer's disease to understand how it occurs and develop ways in which to slow that down and stop it. And in reality, there are no drugs available at the moment which actually slow down the course of dementia. There are a number of reasons for this, but probably the main complicating factor is how complicated the brain is. And unfortunately, it's much, much more complicated than Leonardo da Vinci imagined it many, many centuries ago. And in fact, even Gray's Anatomy lulls you into a false sense of security. The brain is even more complex than it looks. Because when you zoom in and look at the cells that make up the brain, they form incredibly complex, dense networks connecting with one another across the brain. This is an image drawn by Ramon y Cajal, who was a neuroscientist working around the same time as Alzheimer. And it illustrates the beautiful networks that brain cells form. Now, the problem is that there are lots and lots of these brain cells. One recent estimate suggests there are about 80 billion of them in the average human brain. Now, if you imagine that each of those are forming multiple connections with other neurons, other brain cells, the quite literally mind-boggling nature of the human brain becomes apparent. And that's a challenge which confronts us as neuroscientists and geneticists and clinicians trying to understand the diseases like dementia. And in fact, I think it's even more complex than that. Because a disease like dementia, where you lose some of what really makes you aware of yourself, you lose some of the consciousness of being, really goes back to some of the key things that actually make us human. This is an artist called William Untermolen, who was active in the 1960s through to the 1980s in one of his paintings. William was diagnosed with dementia in the early 1990s and undertook a really quite ambitious series of paintings, self-portraits, following his descent into dementia. And these, to me, summarize and are far more eloquent than any series of clinical data notes about exactly how distressing dementia can be. You can almost see his appearance and his impression of himself melting away in these portraits. What I find most shocking is that this last portrait, which is the last he was able to produce, was actually painted 10 years before he died. He was unable to paint for the last 10 years of his life. He was still a functioning human being, but was unable to express himself through the medium of his art, which had been so central to him. And that, for me, really highlights exactly how horrid a disease Alzheimer's is. So what are the missing pieces to the puzzle that are preventing us from understanding what causes dementia and thereby stopping it, finding ways to slow it down? And in essence, it boils down to the fact that we actually understand what happens at the end of dementia quite well. Cells die. The brain loses volume. It loses the ability to function. The problem is that we don't have a clear idea as to what's causing that cell loss, that cell death. And there are a number of ways in which, in the last 10 to 15 years, we've been trying to understand how that occurs. One of those ways is by using genetics, and that's the focus of my research and what I'll talk a little bit about now. And essentially what we do with genetics is we zoom in on the DNA that's found within every cell in your body and that provides a template and a code for how that cell functions and the role it plays within in the body. By looking into the DNA of individuals who develop Alzheimer's, in some cases we're able to identify changes in that DNA which are predictive of developing the disease and therefore predictive of developing the cell loss that we're trying to understand. And the key tool in understanding all of that is actually the simple family tree, and hopefully you'll recognize this as a fairly well-known family. What this highlights, and family trees, your own family trees will do exactly the same, is that you can follow the passage of characteristics, of traits, from generation to generation. If you look at Prince William down here on the left, go back up to Prince Charles and then all the way up to Prince Philip, you can see that they look broadly similar. Some of their facial characteristics are similar. Some of their character traits maybe even will be quite similar. And in fact, all three of them suffer from male pattern baldness. Now, how does this help us understand dementia? So for the vast majority of people who develop Alzheimer's disease, there's no indication of a genetic basis. They don't have any family members who develop the disease as well. But in a small but significant minority, probably about 1 in 10, maybe 1 in 20, they actually have a very, very clear pattern of inheritance. And you can see the disease passing from generation to generation. What this tells geneticists and clinicians is that there's a genetic basis for that disease, that there's a change in the DNA which is resulting in those people developing disease. And so what geneticists and neurologists have been doing over the past 20 years is looking at that DNA, looking at the individual human chromosomes that make up the human genome, searching for the genes that cause disease. And actually we've been very successful in doing that. If you look on chromosome 1, many, many years ago, about almost 25 years ago now, a gene called presenilin, a gene which causes premature senility, was discovered by researchers working in Canada in a family who developed Alzheimer's disease. And having a mutation in that gene predicts almost with certainty that you will go on to develop dementia. If we look at chromosome 21, researchers actually based here in London identified mutations in a gene on chromosome 21 called APP, which is actually the source of that sticky protein that Alzheimer's saw all those years ago. And coincidentally, the reason that individuals with Down syndrome have a much greater risk of developing dementia is because they have an extra copy of chromosome 21. And they have an extra copy of this gene which produces sticky protein. So, in addition to these very, very deterministic genes, where if you have a mutation, you will develop the disease, we're now also beginning to get an insight into more subtle changes in the human genome, which make it more likely for you to develop a disorder like Alzheimer's, rather than not. And actually recently, looking at chromosome 6, Dr. Rita Guerrero and Professor John Hardy, working here at University College, identified a gene that did exactly that. If you had one form of this gene, you were much, much more likely to develop dementia. All told, though, all of these variants account for only a small percentage of people developing dementia. So why are they important? Well, first of all, they give us an insight into the biological underpinnings of this disease, basically what's going wrong in the cells that causes them to die. With the example of the gene on chromosome 6, we think that changes the way in which the human immune system works, causing your brain to become inflamed and causing cells to die within the brain, and that this leads to disease. And that's giving us new ideas as to how we might be able to treat Alzheimer's disease and develop new drugs. But I'd like to give you another example of how this information about the genetics underpinning dementia can help us. And as I said earlier on, we've known for a long, long time that if you develop Alzheimer's disease, your brain undergoes a huge change. And this is illustrated here. Over on the left-hand side, you've got a normal brain. And over here on the right-hand side, you have the brain of somebody who died of Alzheimer's disease. And you can see that huge holes open up in the brain. And that's basically reflecting the loss of cells, the cell death going on in the brains of people with Alzheimer's disease. Now, for many, many years, we were only able to see that at the end of the disease. Basically, when someone had died of dementia, they could undergo an autopsy, as Augusta Dieter did, and the brain could then be examined to see what changes had occurred. And that only tells you about the very last bit of the disease. Now, because we know that certain mutations can lead to disease, we're now able to follow using the advanced brain imaging techniques such as magnetic resonance imaging. We can actually follow that degenerative process over time and follow what happens to somebody's brain whilst they're still alive. And this is important for a number of reasons. First of all, it gives us a chance to have a look and see if there are other changes that are occurring throughout their body, maybe in their blood and so on and so forth, to develop markers that we can use to identify people who are going to go on to develop Alzheimer's disease long before they actually develop it. The other side of all of this is that we can now use patients who have these genetic forms of disease to try out new drugs and actually put them into humans and see if we can slow down that process, slow down the rate at which the brain degenerates. So if Augusta Dieter was living today, would we be able to give her hope? In terms of treatments that are available now, unfortunately not. The situation really hasn't changed all that much from 1906. Certainly, she would be cared for in a very, very different manner. She wouldn't be condemned to an asylum. And huge advances have been made in looking after patients with dementia and making their day-to-day life much, much easier. But I think for future generations of Augustas, people who go on to develop dementia in the future, 10, 20 years from now, I think we're really beginning to see an element of hope. We're beginning to start putting drugs into clinical trials with a real hope that they might actually be able to slow down the disease. And we now have the tools and the patients working with clinicians and with neurologists to actually understand that. And that's actually what motivates me as a scientist to go into the lab every day to try and understand what goes on in these diseases. Hopefully that's given you an insight into how genetics can inform our understanding of dementia. And thank you very much for your attention.